The overall goal of this procedure is to visualize and quantify the differentiation of stromal cells into adipogenic lineage using a lineage-specific protein marker, fatty acid binding protein 4. This is accomplished by first isolating and expanding the stromal cells of interest. The stromal cells are then harvested and plated into standard in vitro culture plates. After incubation for a few days, cells are treated with an adipogenic differentiation medium. Cells are incubated for 14 days with regular media replacement. After differentiation, cells are fixed and stained with antibodies against fatty acid binding protein 4. An automated high-content screening immunofluorescent microscope is used to capture images of the labeled cells in the microwell plates. The differentiation is then quantified using specialized image analysis software.
In contrast to the traditional methods of measuring adipogenic differentiation utilizing dyes such as Oredo, the assay described in this video utilizes an antibody against lineage-specific protein, fatty acid binding protein 4, to confirm adipogenic differentiation. In doing so, this method quantifies changes in gene expression that correspond with adipogenic differentiation. This assay is able to deliver a wealth of information including percentage of differentiated cells, intensity of the fluorescent signal per cell, and changes in cell morphology. The ability to analyze multiple characteristics enables the identification of potential subtle changes in differentiation in response to various treatments. This assay also has high throughput capability, making it ideal for high throughput drug screening applications.